Dear viewers, greetings. In this video, we are going to see about scanning tunneling microscope. In short form, it is called as STM. Scanning tunneling microscope. This STM is a real space imaging technique that can produce topographical images of a surface with atomic resolution in all the three dimensions. And the scanning tunneling microscopy belongs to an expanding family of instruments which are commonly called as scanning probe microscope. The resolution of the scanning tunneling microscopy is approximately 0.01 nanometer. And next is the principles of STM. The STM is based on several principles. One is the quantum mechanism effect of tunneling. It is the effect that allows us to see the surface. Another principle is the piezoelectric effect. That is, it is the effect that allows us to precisely scan the tip with angstrom level control. And lastly, a feedback loop. It is required which monitors the tunneling current and coordinates the current and the positioning of the tip. And the scanning tunneling microscope is a powerful instrument that allows one to image the sample surface at atomic level. As the first generation of scanning probe microscopy, this scanning tunneling microscopy paves the way for the study of nanosciences and nanomaterials. For the first time, researchers could obtain atom resolution image of electrically conductive surfaces as well as their local electric structures by using this scanning tunneling microscope. Next is history of scanning tunneling microscope. Gerd Binnig and Heinrich Roher developed the first working scanning tunneling microscope in the year 1981 while working in the IBM Zurich Research Laboratories, Switzerland. Binnig and Roher chose the surface of gold for their first image. When the image was displayed on the screen of the television monitor, they saw rows of precisely spaced atoms and observed broad traces separates by steps one atom in a height. And the Binnig and Roher has discovered in the scanning tunnel microscope is a simple method for creating a direct image of the atomic structure of the various surfaces. And the discovery of Binnig and Roher opened a new era for science surface, and their impressive achievement was recognized with the award of the Nobel Prize for Physics in the year 1986. Mode of action of scanning tunneling microscopy. The scanning tunneling microscopy can be operated in two modes. The first mode is constant current mode. The second mode is constant height mode. This slide shows the first mode, that is the constant current mode. In this constant current mode, the current is made constant during the scanning by changing the distance between the tip and the surface. Then the second mode is, this is the second mode, that is the constant height mode. In this mode, the tip height is made constant and tunneling current at every step of scanning is measured. In the first method, tunneling current is constant. In the second method, that is the constant height mode, the path of the tip is constant. This slide shows the working principle of scanning tunneling microscope. And the main component of the scanning tunneling microscope is a rigid metallic probe tip which typically composed of tungsten, and this tip is connected to a piezo drive which contains three perpendicular piezoelectric transducers. The tip is brought with a fraction of nanometer of an electrically conducting sample. At close distance, the electron clouds of the metal tip overlap with the electron clouds of the surface atom. If a small voltage is applied between the tips and the sample of tunneling, the current is generated. The magnitude of this tunneling current is depending on the bias voltage which was applied and the distance between the tip and the surface. A current amplifier can convert ungenerated tunneling current into a voltage and it is passed to the distance control and the scanning unit, and finally it reaches the data processing and display. Finally, the information is gathered by monitoring the current according to the tip position, which scans across the surface, and this information is usually displayed in the form of image. This image shows the silicon surface atoms, that is enlarged 20 million times, color enhanced by the computer. This is the world's first image of the individual surface atom, and the bonds that hold them in a place were produced by research technique developed by IBM scanning tunneling microscopy. This image shows the scanning tunneling microscope image of a gold surface. In this image, the individual atom and their arrangements are directly visible. And here, dark bands correspond to the pits in the surface with the absence of atoms are observed. This image shows the scanning tunneling microscope image of a single cobalt atom on a copper surface. This image shows the scanning tunneling microscope image of a quantum coral of cobalt atoms in an ellipse on copper. Next is application of scanning tunneling microscopy. This scanning tunneling microscopy helps the scientist to get a picture of how the atoms are arranged on a surface by looking at the electron density of the surface atoms. This scanning tunneling microscope is used to image the topography and measures the surface properties, manipulates the surface texture and surface roughness for initiating the surface reactions of the various reactions, and the shape, size and organization of an individual particles or molecules along with the topographical information can be determined by this scanning tunneling microscopy. And electronic information of various conductive surfaces can be determined by scanning tunneling microscopy. And in material science, it provides new insights into the nanoscale properties of known materials and enables the study of new nanoscale materials such as graphene and carbon nanotubes, as well as assembling the structures composed of an individual atoms. In chemistry, the scanning tunneling microscope allows how the surface roughness and electronic properties of catalysts govern their performance to be understood. And finally, many biological samples are not electrically conducting. It has been shown that they can be coated with thin metal films which are deposited on the conducting substrates or scanned under humid conditions so that they can be studied by using the scanning tunneling microscopy. The next is advantages of scanning tunneling microscope. And the scanning tunneling microscope is capable of capturing much more details than the normal laser microscopes. It also helps the researchers for the better understanding of the subject of their research on a molecular level. And these scanning tunnel microscopes are also versatile. They can be used in ultra high vacuum, air, water and other liquids and gases. And this STM will be operated in temperature as low as 0 Kelvin up to a few 100 °C. The next advantage is high resolution image, that is in atomic scale, low power application, and finally no damage to the samples. Finally, limitations of scanning tunneling microscope. It includes difficult to use effectively, and it requires a lot of skill and precision. It requires very stable and clean surfaces, excellent vibration control and sharp tips, and it uses highly specialized equipment that is fragile and expensive. It needs for vacuum and vibration isolation. The samples limited to conductors and semiconductors, high equipment cost, surface preparation, and maintaining the tool sharpness is very tough. These are all the limitations of scanning tunneling microscope. Dear viewers, thank you for watching and thank you for your support. Thank you.